Inside all of our cells, we have mitochondria. And inside the mitochondria, we have a matrix where a cycle of reactions occurs. That cycle is called the citric acid cycle, or the Krebs cycle. During this cycle, energy from food is taken and made into something that cells can use. Before the Krebs cycle can do its thing, pyruvate, which was made during glycolysis, enters the mitochondria where it is turned into acetyl coenzyme A by first removing pyruvate's carboxyl group, leaving the remaining to be oxidized, forming acetate. Coenzyme A then attaches to acetate via its sulfur atom, and acetyl coenzyme A is formed. Let's take a closer look at the citric acid cycle. The citric acid cycle can be divided into 8 different steps, each with its own enzyme, resulting with 1 ATP, 3 NADH, and 1 FADH2. In the first step, acetyl coenzyme A, that came from pyruvate oxidation, takes its 2 carbon acetyl groups and adds it to the oxaloacetate, which produces citrate, or citric acid. In step 2, one water molecule is removed and another is added to convert citrate into isocitrate. In step 3 of the citric acid cycle, isocitrate is then oxidized, reducing NAD to NADH. The remaining compound then loses a carbon dioxide molecule. Eventually, it becomes ketoglutarate. In step 4, another carbon dioxide molecule is lost. The remaining compound is oxidized, reducing NAD plus to NADH. Coenzyme A is then attached to the remaining molecule by an unstable bond, becoming succinyl coenzyme A. In step 5, a phosphate group displaces coenzyme A, which is then transferred to GDP, which forms GTP, a molecule that is similar to ATP. GTP can also be used to generate ATP. The enzyme that is left is succinate. In step 6, 2 hydrogens are transferred to FAD, which form FADH2, an oxidized succinate. The enzyme that is formed is fumarate. In step 7 of the citric acid cycle, the bonds in the substrate are rearranged when a water molecule is added, which forms malate. In the last step of the citric acid cycle, the substrate is oxidized, reducing NAD+ to NADH and regenerating oxaloacetate.